Hello everyone, this is Redam Rabbit from Group 5 and today our topic is the Mendelian genetics. So the Mendelian genetics explains how traits are passed from parents to the offspring. Also, Gregor Mendel discovered that some traits are dominant and the others are recessive, and they follow a clear inheritance patterns. Also, the Mendelian genetics is the basis of the modern genetics.
Hi, we are going to talk about the Mendelian genetics, the dihybrid cross in pea plants. This research investigates Mendel's law of independent assortment through an experiment involving pea plants, the same model organism Gregor Mendel used in his groundbreaking studies on heredity. In this investigation, a dihybrid cross was conducted to explore how two traits, seed colors and seed shape, are inherited simultaneously. The goal was to determine whether these traits segregate and assort independently during gamete transformation. As Mendel law predicts, the expected outcome was a 9:3:3:1 phenotypic ratio, representing the combinations of yellow round, yellow wrinkled, green round, and green wrinkled. All of these seeds, after collecting and categorizing, was applied to compare and observe unexpected results. This analysis helped determine whether any deviations from the predicted ratios were due to random chance or other factors, ultimately reinforcing the reliability of Mendel principles in explaining genetic inheritance and patterns.
With our introduction, Gregor Mendel's pea plant experiments help scientists understand how traits are passed from parents to offspring. His work introduced two main rules of heredity: the law of segregation and the law of independent assortment. The law of segregation explains that each parent gives one gene for every trait, with the law of independent assortment says that the different traits are inherited separately. In this study, we focus on how two specific traits, seed color and seed shape, are inherited together in pea plants. By crossing parent plants that are heritageous for both traits, we can predict the possible outcomes for their offsprings. Mendel's theory says that these offspring will show certain patterns or phenotypic radios that match his genetic laws. Our goal in here is to test if our results follow Mendelian radios by using a chi-square test to analyze the data. This statistical test helps us see if what we observe in our experiment matches what we expect. Through this experiment, we can better understand how genes work, how traits mix, and why Mendel's discoveries are still important in modern genetics.
This is our methodology. Our prepared materials are pea seeds, trays, tweezers, magnifying lens, Punnett square templates. Our identified traits are seed color and shape. Crossed heterozygous parents constructed a 4x4 Punnett square showing gamete combinations, recorded seed phenotypes and counted each category, applied chi-square test to compare observed versus expected ratios, interpreted significance.
The results or preliminary results that we have for the phenotype, yellow and round, we had expected a ratio of 9, and the expected percentage was 56.25%. Yellow wrinkled, expected ratio is 3. The expected percentage, 18.75%. Green and round, expected ratio 3, and expected percentage was 18.75%. Green wrinkled, expected ratio is 1, and expected percentage is 6.25%.
Our conclusion or expected outcomes. The experiment is expected to produce a 9:3:3:1 phenotypic radio, which support Mendel's law of independent assortment. This ratio showed that the two traits seed color and seed shape are inherited independently from each other. Each combination of alleles occurs based on change, proving that genetic inheritance follows predictable patterns. Although we expect results close to the 9331 ratio, there might be small differences due to random variations, counting mistakes, or limiting in sample size. These differences are normal and remind us that probability plays a key role in genetics. This experiment demonstrates how genes separate and assort independently during the formation of gametes, creating genetic diversity in offspring. Overall, this study helped us understand the importance of Mendel's discoveries in modern biology. Using AI tools was helpful in organizing ideas, improving the clarity of explanations, and ensuring that scientific terms were used correctly. It made the process of writing and analyzing easier, but the main understanding and interpretation of the experiment came from learning Mendel's principles and applying them to real data. Over here we have the comparison in the graph that we have over in here. That the yellow round 56 shows a comparison of 28.1. The yellow wrinkle shows like 5.6. The purple wrinkle showed 22.2% and 11.1%. Yellow smooth 22.2, 11.1. Purple smooth 44.4%. Green wrinkle 6.25% and 3.1%. Green round 18.75, 9.4%. Yellow wrinkle 18.75 and 9.4%. And that's it.
Thank you so much for watching our video.